Ẹ jẹ́ padà wá later.
Good morning, everybody, and you're welcome to another Friday morning coffee, as we popularly call it. Again, this is our regular or weekly intensive research training hosted by the Central Office of Research in collaboration with the Postgraduate College and the Postgraduate Student Association at the Obafemi Awolowo University, Ile-Ife, Nigeria. And today's topic is the technicalities of field experimentation and data analysis 2. This is a second part to this particular series taken by Professor Richard Akinwale. But before we start, I would like to call on the Deputy Vice Chancellor, Research, Innovation and Development to give us an opening remark or to open up this. Thank you. Prof, over to you, sir.
Thank you so much, Toby. Again, let me say good morning to all. Again, welcome to today's. Hello, please, can you hear me?
Loud and clear, sir.
Oh, okay, good morning, all. Yes, please. Again, welcome to today's workshop, the one we call the early morning coffee. Good morning, and again to welcome us to this. Again, to thank those who have constantly been part of this program, because without the audience, it will be difficult to continue to do this. So let me again welcome Professor Richard Akinwale. If you remember, he gave us a half cup of coffee about 3 weeks ago. So I think this morning now he has come with the full package, the remaining half, and I'm sure.
I think we lost him.
Yeah.
For early career people, or even those of us who are in our mid-career, this is not just about listening to lectures. It's also a good opportunity for us to connect and also to network with ourselves. So again, welcome, Prof, and thanks to all. Over to you, Toby.
Toby, over to you.
Professor Odu, I think you can take the floor. I think maybe Toby is busy.
So, good morning, everyone. You are welcome to another time, another Friday that is being slated for the duration of one of our seminar series. So, this morning we have Professor Richard Akinwale, who is going to present to us technicalities of field experimentations and data analysis. So, this is especially a good topic for us who are in the agricultural sector, especially those who have field experiments. So I want to ask you to just listen and then prepare your questions as the presentation goes on. I want to welcome Professor Richard to give us this presentation this morning. Thank you, Prof. You can come on. You can start now.
Okay. Okay, good morning. Good day, everyone. I hope everybody can hear me. Hello?
Yes, Prof. And yes, we can hear you, sir. Can you put your slide on slideshow, sir?
Yeah, it's already on
slideshow.
I am already sharing. Yes, it's on slideshow already.
No, put it on slide.
Okay. Okay. Where are you? Okay, can you see?
It's good now. It's very good now, bold. Okay.
Very good,
Thank you.
Prof. Yes.
Oh, thank you very much. Once again, good day, everyone. I'm happy to be here again to give the remaining half cup of the coffee I gave a couple of weeks ago. I want to say a big thank you to the COR and Postgraduate College for this innovation. It is very apt. Yes, straight to business. We want to conclude the business we started a couple of weeks ago on technicalities of field experimentation and data analysis. Today is part 2. As a way of recap, I started discussing why field experimentation in the last edition. That field experimentation is very important because that is actually the final target. It is on the field that our farmers practice, not in the lab, not in the screen house, mostly, especially in sub-Saharan Africa. So all our innovations has to get to the field for the field to test whatever good results we got from our lab or from the field. So we talk about the peculiarities of field, how several factors are acting and interacting to affect the treatment that we are applying, and how we need to take care of these factors so that they will not confound the results. We talked about guard rules, border rules, some important technicalities or terminologies in experimentation. We discussed the principles of experimentation generally. We discussed overview of experimental designs. So this morning we're going to be talking on the experimental designs, the peculiarities of the experimental designs, how the analysis is carried out on them, and the application of these principles and statistical principles to crop improvements, how we have used these principles in developing some materials that has bettered the life of farmers and consumers, especially in sub-Saharan Africa. So I'm going to begin by discussing the complete block designs, which are designs that informs all treatments being present in a replicate or in a replication. So an example is completely randomized design. The assumptions under completely randomized design is that there are no extraneous factors that can introduce variation. The area where the experiment is carried out is so uniform that it is only the treatment that will vary the level that is responsible for variation. But in most cases, there may be so many sources of variation in your experimental area that you don't capture if we're using this completely randomized design. And that is why it is not suitable for field experimentation. It could be used for laboratory experiments where you are carrying out a test tube experiment. You know the reagents, you are varying the levels, you know the medium you are using, and you can control virtually everything and even the treatment you are applying. So that design could be used, but certainly not in the field. The second type is the randomized complete block design. Randomized complete block design. The assumption under this is that we agree there is variation in the experimental area, but that variation can be partitioned. If there are 3 or 4 sources of variation, we can identify those sources of variation and consider each source as a replicate. If we have an experiment where the soil type is like into 4, So we can say the soil type 1 is rep 1, soil type 2 is rep 2, soil type 3 is rep 3, soil type 4 is rep 4. If we are in a screen house where there is different degrees of temperature, we can decide to partition the degrees of temperature into 1, 2, 3, 4. So each degrees will be a replicate. So that is how randomized complete block design is captured. Within each replicate, you have all your treatments present. Rep 1 contains all the treatments, Rep 2 contains all the treatments, Rep 3 and for Rep 4. So it is regarded as randomized complete block because all the treatments are present in each block. So it is possible for you to run the analysis and the block effect is significant. It is not a disadvantage because you have already blocked for variation that might confound the result of your experiment. So randomized complete block design is appropriate for any field experiments, very appropriate for field experiments. We can have some other situations where randomized complete block design might not adequately capture that variation, and we use what we call Latin square design. In Latin square design, within a rep, we might not be too sure that there is no variation or there is variation. And so we divide or we partition the rep effect into the colon effect and the row effect. In so doing, the degree of freedom that is attached to replicate is subdivided into row effect and the colon effect. And it makes the experiment to be more, the analysis to be more robust. Then we have this second group of field experimental designs that we call the incomplete block design, or what we call the lattice design. This incomplete block design is used when the level of the treatment we are applying is very, very large. So large that, again, we cannot be sure that there is uniformity within a replicate. So if we are sure there is no uniformity within a replicate, then we need to subdivide each rep into a smaller block that is called block within rep effect. And so this block within rep effect captures the variation that might exist within a replicate. And so within the block within rep effect, it is not all the treatment that is present, but eventually all the treatment are present within a rep. So there is an additional factor that we're going to introduce into the analysis of variance that is going to capture that within block effect. And when we are computing the means will not be arithmetic means as we have it for complete block designs like randomized complete block design, lattice square designs. We are going to use the arithmetic mean. That is when you say 2 plus 3 divided by 2 would be equal to 2.5. But in incomplete block design, we need to adjust. That is a factor that we're going to use to adjust for the incomplete block, for the block within rep effect to give us the right means, which is the adjusted mean that we will use for the incomplete block. So incomplete block design is divided broadly into two. We have the balanced lattice design and we have the partially balanced lattice design. The partially balanced lattice design is also referred to as the alpha lattice design or the rectangular lattice design. We also have experiments where we are considering two or more factors simultaneously. Any of these design can be used for when we are having two or more factors that we are studying at the same time. And when we're having these two or more factors, it means that we are interested in the main effect of the factors and how the factors interact, how one factor, when present, affects the expression of the other factors. So we also have some designs that we use when we have factorial experiments like this. We can use randomized complete blood design for the factorial experiment as earlier explained. We can use a split plot design. That is when the factors we are considering will require different precision levels on each factor. So the ones that we require more importance, we attach more importance to, we consider it as a subplot factor, and the one that we don't attach more importance to, we consider it as the main plot factor. And these are some of the layouts for the different experimental designs I have discussed. For complete randomized design, when we have 4 treatments and we have 4 replicates, this is how it looks like. There is no particular pattern of distributing the treatments. You just distribute them randomly. So each treatment has a fair chance of falling into any experimental unit. So this is how it looks like. And remember, I said it's not appropriate for a field experiment. And then this is the layout for a design that we consider as randomized complete blood design. We also have 4 treatments, treatment A, B, C, D, and we have 4 replicates as it is divided. So we have 4 times 4, 16 experimental units here. So if you look at it from the top, in the first rep, all the treatments are available. Treatment A, B, C, D. In the second, you also have treatment A, B, D, C, D, but randomized. So A is in block 3, is in plot 3, B is in plot 1, C is in plot 2, D is in plot 3. In the third rep, the same thing, that is randomization, and in the fourth rep, there's randomization. So in each of the experimental design, the principles of experimentation are obeyed, which means randomization, the experiments are well randomized, and the experiments are well replicated. Then there must be one of the treatment that is going to serve as the control, or that is going to serve as the reference point in it. So this is how randomized complete blood design looks like. Then when we talk about lattice design, the principle of the layout is that in each of the replicates, you have a treatment appearing once in both row and column. So if we look at the first row and the first column, we will discover that treatment A appears once. If you look at the second row and the second column, treatment B appears once. The same thing for third column and third row, the same thing for fourth column and fourth row. So this layout is very important when we really don't know the pattern of distribution of the soil gradients or the fertility gradient of the soil. When you can't say, oh, the soil is such that at the upper part, the distribution of fertility is lower compared to the lower parts, you don't even have any idea. That is when we use the Latin square design. Then we have some of the layout for the factorial experiment when we have 2 factors. If we are considering 2 factors, when we have a factor, the 2 factors, and we are interested in the interaction of the 2 factors, even above the effect of the 2 factors. So we have the layout we call the strip plot design. Strip plot design. It is a bit different from split plot design, because in split plot design, we're interested in the in the subplot effect. We are placing more emphasis on the subplot. But in the strip plot design, we are placing emphasis on the interaction of the two factors rather than the main factors. So we use strip plot design. Then we have split plots, and this is how the split plot is. You have your main plot effect, and within each main plot, you have the subplot factor. In this case, we're using N to represent the nitrogen level. So we have 3 nitrogen levels, and we have about 6 varieties within each nitrogen level. So you have nitrogen level 1, which is the main effect, and you have the varieties, 6 varieties that are present in each of the nitrogen level. So we are considering the nitrogen level as the main factor, and we are considering the varieties as the subplot factor. So this is an example of a split plot design. We have some of the analysis of variance table model for each of the experimental design. This first one is the source of variation, is the analysis of variance for completely randomized design, where we have under source of variation just 3 factors. You have your treatment, then you have your error, then you have your total. In this example, where we have 5 treatments and 4 replicates, we have given how the degree of freedom is distributed. So for 5 treatments and 4 replicates, we are going to have a total experimental unit of 20. So the degree of freedom for total will be N-1, which is going to be 19. The degree of freedom for treatments, since we have 5 treatments, will be 4. Then all other things will go inside error. So the error looks big when we consider the proportion of the treatments to the proportion of the error as regards the total. So it looks big. But when we use randomized complete blood design, the scenario changes a bit. We have, instead of 3 sources of variation, we have 4 now. We have replicates. Since we have 3 reps or 4 reps, so we have replicate effect, we have treatment effect, we have error effect, and we have total. The total does not change. It is still a degree of freedom of 19. But the error changes from 15 to 12 because the effect of rep, since we have 4 reps and the degree of freedom is 3, that 3 is taken out of the error. So it makes the precision of these experiments or this analysis to be better than that of completely randomized design, because any model, any statistical model that reduces the error is actually more effective than the one that allows the error to be big. Remember in our last class, I said the error is like the noise. So any means of reducing the noise will magnify the information that we are passing across. So exactly that can be applied to this. Then for the Latin square design, I have told us that the rep effect is partitioned or subdivided into the row effect and the colon effect. So the row effect, we have 4 rows, and the colon effect, we have 4 colons. So each of the source of variation takes 3 degrees of freedom. So that reduces our error further from 15 in CRD and 12 in RCBD to 6 in Latin square design. So this design is also better because it gives it reduces the error degree of freedom, and it makes the experiment to be, or the analysis to be, more robust. Then we have this factorial setting when we have 2 factors. I have given an example here. We have treatments and we have variety. So when we have 3 treatments and we have 5 varieties that we are testing, this is how the analysis of variance table would look like. Outside the replicate, we have the treatment effect, which is the factor A. We have the variety effect, which is factor B. And we have the interaction between factor A and factor B, which is the interaction effect. So for a randomized complete block design in the factorial arrangement, we are interested in everything, both the main effect, which is the treatment and variety, and the interaction. So we are placing equal emphasis on the 3. So this is how the analysis of variance table will look like. Then for a split plot design, where we have more interest in a factor over the other factor, I have used A and B as the factor of the experiment. So the peculiarity of this split plot design is that we're not going to use just one error to test all the effects, because we are more interested in one factor than the other. So the pooled error that we used in the previous table 4, we are going to subdivide it into the error A, which is the main plot error, and the error B, which is the subplot error. So if we have error like, let me give this example. We have degree of freedom of 28 for the pooled error in table 4. That pooled error will be subdivided. Smaller part of the error will be used to test the main plot factor, which is going to be our error A in the 4th row here. Then the bigger error will be used to test our factor B, which is the subplot factor, as well as the interaction factor. So this is how the layout of a split plot design looks like. Then table 6 gives us a picture of an incomplete block design or a lattice design. This is how the incomplete block design analysis of variance table looks like. Apart from the rep effect, we are introducing another factor, which is called the block within rep effect, as I've earlier explained. The block within rep effect signifies that within a rep, we have smaller blocks. Within the smaller blocks, there is uniformity, but in the larger rep, there may not be uniformity. So that effect is called the block within rep effect. It has to be created, then we have our rep effect, then we have our treatment effect, then we have our error effect. I have done this for a 25 treatment levels. When we have a treatment that has 25 levels, 25 is considered to be large for a field experiment, because we can be sure that where we lay treatment 1 will be the same as where 25 treatment will be. So we need to create that smaller blocks within the rep. And so this is how the analysis of variance table will look like. The use of adequate experimental design actually defines the acceptance of the result of the analysis that is carried out. This analysis of variance, we are very, very much interested in it, because it helps us to adequately analyze the effect of the treatments that we are applying without the compromise due to extraneous factors. If the design is wrong, everything about the experiment is wrong. And we can imagine if we have an experiment that is sponsored, we have used our time, we have used our funding, we have used our resources, we have used the land, so many things we have used. And if the design is wrong, everything is wasted. Everything is wasted. It is like going back to the field to carry out the experiment again. So the knowledge of experimental design is very, very pertinent in analysis, because I have come across some running analysis without even knowing what the experimental design is. Don't have the knowledge of the experimental design. So I am a plan builder, basically, and on the basis of these experimental designs, statistical analysis, we have conducted a lot of experiments, and we have developed a lot of maize varieties, maize hybrids, maize inbred lines that are fantastic, that are superior to the existing one, especially in sub-Saharan Africa. How do we do it? We have developed maize that are tolerant to some of the biotic and abiotic stresses. Biotic and abiotic stresses, I mean stresses like drought, when there is a shortage of rainfall or shortage of water supply. We have developed some varieties on the basis of these statistical principles. We have developed varieties that are tolerant to this parasitic weed that we call striga, which is prevalent in the Savannah region of Nigeria, which is actually the maize belt of Nigeria. We have developed materials that are tolerant to poor soils. Generally, most of our soils in the tropics, they are very, very fragile and they are poor. Many parts of our soils are acidic, and so it reduces the productivity of farmer. But in maize breeding, by collaboration with scientists from IITA, we have developed a lot of materials that are tolerant to low soil fertility, especially low soil nitrogen and soil acidity. We have developed materials, and all these are on the basis of the application of right statistical principles. So in short, what we do in applying statistical principles to crop improvements is to help us to study phenotype. What I mean by phenotype is what we cherish in the plant: yield, resistant to pests and diseases, tolerant to abiotic factors like drought. We have been able to use statistics to partition the phenotypic traits into the genotype, the environment, and the genotype by environment interaction. What we're interested in plant breeding is actually the genotype component. So, but from phenotype, we were able to identify the genotypic effect. And not just that, we have partitioned the genotypic effect into its components, to the additive gene effect and the non-additive gene effect. We also partitioned the non-additive gene effect into its components, which are the dominance effect and the epistatic effect. We also partitioned the epistatic effect into the different types of epistasis. So all these are used in bringing about genotypes or bringing about varieties, hybrids that are very, very good, that are very superior to the existing varieties, And we have recommended a number of these to farmers, and the farmers are planting them. Well, there are still a lot of them that are still under study, which we are thinking of introducing to farmers in subsequent years. So what I am saying now is based on these principles I have explained and the statistical analysis, we have used analysis of variance to bring about better varieties from the existing varieties. We have used regression and all the types of regression to predict which is applicable to what we call precision agriculture. That when you plant this variety and you have these factors in the field, this is the yield you should be expecting. It has been on the basis of regression and correlation and some other statistics. We use factor analysis, we use part coefficient analysis, we use cluster analysis, and some other analysis, statistical analysis to bring about this fit. So in conclusion, I want to say that adequate knowledge of field experimentation and statistical analysis is basic to improvement in research. Then if there are no good design, then there is no valid results. So time, funds, energy that is put into any experiment is going to be a waste if we don't have adequate knowledge of field experimentation and statistical analysis. So it is important to have adequate knowledge of experimental design so that our results can gain global acceptance. Thank you very much for your attention. Over to you, sir.
Thank you very much, Prof.
Okay.
I need to confirm if you all can hear me.
Yes, I can hear you clearly.
He's done. Okay. Maybe I can take over. Thank you, Professor Akinwale, for this presentation.
Thank you, sir.
It's something that I understand very well, and I believe that all that you have presented are applicable in other areas of endeavors.
Yes, yes.
So, as a biometrician, you are available, I presume, for people to contact you from
Yes,
anywhere on campus,
yes,
from even outside
university.
campus. After university, you are available.
Yes, sir.
So I want
Yes,
to ask that questions be asked. If you have any questions, can you just raise up your hands and then, or if you need any clarification on what has been presented, any aspect of it, so you can ask, please. Questions? Questions? Nobody's ready
Sorry,
to.
Professor Odu.
Okay, yes, sir.
Sorry, I don't know how many people who are here who are not particularly from that research bias, who are not from agric or particularly from crop breeding. And because
okay.
I followed the presentation for me, and I think it was very, very rich. So I would really want to challenge people listening to Prof. Are you able also to bring this into the broader context of experimental design? Because I was just trying to listen to him, and I was saying exactly that experimental design, that clinical practice, social sciences
Okay.
or humanities, I think is almost the same. So I
Okay.
really want to encourage those who are not in this field to see, how that was also adapted, within this
Okay.
field. Yeah.
Okay.
But Professor Akinwale, for us, I did a lot of work in global health, and a major challenge we have when we are reviewing proposal or we are writing this is how do you contain, either in terms of the design or in terms of the metrics, analysis, etc., issues around diffusion and also issues around contamination. Thank you.
Okay. Right. diffusion and contamination. Okay. Okay, sir. Should I answer right away or?
Yeah, go ahead and answer that, and then somebody's raising a hand. So maybe when you answer that, and then we can allow the next
No,
person
okay.
to ask our own question.
Okay, all right. Diffusion and contamination. Well, maybe that might not be applicable to us directly, but if I get it right, I think you're talking about outliers when you are collecting data and some of them falls outrightly off what you expect. How do you handle it? Well, if that is the case, there are provisions in statistical analysis that can help you to identify outliers and will compute a value that is more appropriate than those values that are outliers, which is not going to make your result to be very weird and not realistic. So this is handled especially when we use the type 3 sum of squares, which is more friendly to data that are off point. It is going, or if we have some experiments where the data is not balanced and you have outliers, it is going to handle those very well. It is going to compute some values that can replace the outliers that is going to make your results more acceptable or more realistic than putting those contamination, then your results. And when the analysis is done, you'll be wondering, where does this one come from? So if that is what you mean by contamination and diffusion, I think that is how we handle it in our side. But if it's not, maybe you can throw more light on it. I can also.
Sorry, I mean, I'm just trying to bring, so for experimental design generally,
Okay, okay, sir.
the whole concept is to say that we are exposing a group, we are
Okay,
taking this other group that we are not exposing. So there are
okay,
different possibilities in those processes when we do our design.
okay,
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 so for instance, contamination means that how do you make sure that the intervention that you have proposed are just the entire intervention that this group are exposed to, and that there
okay,
is no other things that comes in
okay,
between. So
okay,
diffusion talks about, for instance, now you are doing this in your home field. How do you contain that whatever thing that you are doing does not spread, to this other field that you do not really
Right.
intend?
Okay.
So those are
oh,
the kind of contamination and
okay,
diffusion. So both in the
oh,
design and both in the analysis, we look into those
okay.
questions. Yeah.
Okay, okay, sir. Okay, I think the questions are related more into the ethical issues, uh, uh, maybe than experimental. But for the aspect of the experiment, we have something that is closely related to that in agriculture too. When you are applying a fertilizer or any chemical, and there is an adjacent plot, you don't want the chemicals we have applied to this plot to extend to another plot. So that is what I explained earlier on about the border
Blogging.
rules blocking. We introduced a border rule such that even if there will be diffusion or the treatment you are applying here, if it is going to extend, it is not going to get to the next plot where you are not applying the experiment because you have left an adequate gap that can absorb that thing so that it's not going to affect the other plots where you don't intend to apply the treatment. So we leave an adequate gap. But in a health issue, I don't really know how that is handled. For the contamination, it is like the error, the experimental error. And I said the experimental error, there are two things that contribute to it. We have the systematic error, which is actually the mistakes of the experimenter. You are not getting a reading correctly, and you are recording it. So it might or you want to record 2.0 and you mistakenly recorded 20. There are a source of contamination to the analysis. So essentially, the systematic error must be reduced to the barest minimum. And one of the reasons to reduce it is this blocking. When you introduce blocking, you reduce it to a larger extent. Then the experimenter also must be conscious of that fact, that if he is not careful enough, there is a tendency that he introduces a factor that the statistical model will not be able to capture. So that consciousness, when it is in the experimenter, then it will be able to watch or keep watch so that he is not going to fall into that trap. I think this is how best I can relate to the question, sir.
Thank you. So, can we ask Osheshan Obukola to ask our questions now?
Okay.
You can come on, Osheshan Obukola. Come on. What's the question, Osheshan Obukola? Ask to mute. Unmute yourself, please.
Aha. She's
Unmute
still
yourself.
muted.
Okay, let me see if she needs to be unmuted.
She's still muted.
She's muted. Any other question apart from Osheshan Obukola? Okay, let me see.
Okay. Another hand is up, sir.
Yeah, I've seen the hands. Toby, can you unmute those two people, please? Bukola, unmute yourself. Maybe you can post your question then.
Yeah.
If you can unmute. Yeah, can you unmute yourself to Professor Aule? Professor Aule, can you unmute yourself too? If you cannot, then please post your questions. Post your questions, and then let's let the presenter answer them. Can you post your question now? I think they are not able to, so yeah, maybe. Okay, I think they are unable to, and we still have time for everyone. Okay, their questions are already on the chat. Okay, let's see. Let's see the questions. Their questions. Olufade, okay. Olufade is asking if someone is working on bed bugs in Southwest Nigeria, for example, which experimental design is most suitable to minimize error? RCBD, split plot design, or Latin square design?
Well, good question. It depends on the objective that you are trying to achieve. It is not every experiment that requires experimental design. There are if you have no factor experiments, for instance, you are doing a survey, you may not apply any experimental design. But when you are applying a treatment to control the bed bug, you are collecting the bed bugs maybe from different locations and you are comparing. When the issue of comparison and when it comes in, then you have to introduce an experimental design. Well, it depends on what you consider as the factors of this experiment. That is what is going to give us an idea of the experimental design to use. So you may need to provide more information about your experiment so that we can suggest the adequate experimental design to use. Thank you.
Yeah. Maybe before any other question comes, how do you attend to missing data?
Okay,
How do you account for missing data?
okay. I have said it earlier. There are statistics that can help us to estimate missing data based on the data we have collected on um, other replicates. For instance, if you have 4 replicates and you have 5 treatments and you have collected data on 3 of the 4 replicates and some of the treatments you are missing their data in the 4th rep. If you adequately use analysis of variance and you specify for type 3 sum of squares, it is going to provide an estimate of the missing data and run the analysis, and the analysis will be reasonable. So there are statistical procedures in handling missing data. A lot of calculation goes into that I may not talk about here, but we have statistical procedures that handle missing data or handles data that are not balanced. If data is not balanced, so there are procedures, statistical procedures to handle them. But that does not mean that we should be very lazy and collect a data that is not analyzable, just data that has a lot of missing data. That may not, the statistics might not adequately handle that. But if we have a reasonable number of missing data, then we have statistics that can handle that. Thank you, sir.
Okay, thank you. The question from Osashono is, does any analysis package depend on the field layout design used like SPSS? I hope you
Yes,
understand the
definitely.
question.
Sir,
Okay.
yeah, then participant can unmute and speak now, sir. Thank you very much.
Okay.
Buki, can you unmute and speak so that's
Yes,
last question?
yes, sir. Thank you, Prof, for the presentation, sir. So that question that I put on the chat, I said, does the analysis package to use for the analysis, does it depend on the experimental design used? Maybe the SAR, SPSS, or HAR, or we can use any one. Then another question, sir. You didn't talk about some layout like a split plot design. Which
Okay.
I would like to talk about it, reasons to use it, and like that.
Okay.
And the third question is for like a split plot design. What of during layout there is a mix up on the main plot and the subplot? Maybe what you supposed to put in subplot, you put it in the main plot during the feed layout. Is there anything that can be done? Maybe done in analysis, or there's nothing that can be done. You just have to go back to the layout again.
All right.
Thank you, sir.
Okay. Yes. Your first question. You need an adequate knowledge of the statistics that you want to use, regardless of the package you want to use. You are the one to give whatever package you want to use a command. If you want to run analysis of variance based on randomized complete block design, give SPSS the command, it's going to run it. Give SAS the command, is going to run it. Give R the command, is going to run it. I am used to SAS, Statistical Analysis System, GenStat, and R, much more than SPSS and other versions, because it is more applicable to those of us in applied sciences, especially in agri. And I have been used to the result layout, how the result comes out. So, but any other software can be used as long as you know what you want. And when the result comes out, you can say, oh, this, you can relate with the result vis-a-vis the data you have given to it. So any software can be used. That is the first question. Yes, the split plot design. I just talked about the basic ones when we have two factors, which is an example of a factorial experiment. The split plot, we're having three factors. It makes the design to be more complex. It makes the analysis to be a bit more complex, but it can also be undo because you have three factors. So you need the first factor to be the main plot, the second factor to be the subplot, and the third factor to be the subplot. So the same principle that applies to split plot is the one you are going to use for your split plot. And you do it appropriately. About the mix up, if that is mixed up, you have to go back. There is no two way about it because you must have set your objective that, okay, my objective, this is what I need for this. So if you make a mistake and you replace what is supposed to be your main plot to be the subplot, there is no how you can get the same result, no how. So it is better to go and repeat the experiment, except you want to change your objective, change the order of the one that you need precision on. If you change it and it still makes research sense, then you can go ahead and you don't need to go to the field. But if changing it will change the whole thing, it is better to go to the field and avoid this mix-up we are talking about. Thank you.
Thank you, Prof. We have 3
right.
minutes. I think you can look at this question. There is Prof, I came across a design called 2x2 plus 1 control. The person is asking, how acceptable is this kind of design?
This is not a design.
Aha.
It's not
So
a
it's not a
design.
design.
2x2 what? Well, if you have a factorial experiment, you can have a 2x2 factorial experiment that you lay out using designs. You can use CRD, computer randomized design. You can use randomized complete plot design. You can use split plot. You can use strip plot design. So you can use any design for this. But this +1, which is the control, I don't know where it is coming from because this is going to be 5 treatment levels. And when we have 5 treatment levels, you can just use simple randomized complete plot design for the 5 treatment levels. It is still small enough for randomized complete plot design or for complete plot design to undo. So what you have put here is not actually a design. It is an arrangement, a factorial arrangement. But that one control should be lumped inside one of the factors so that because control cannot stand alone. Thank you.
Thank you, sir.
I've been able to see if I can do that in a few seconds, Prof.
All right, sir.
All right. Thank you so much, Professor, my prof, for your beautiful presentation. That's a very good one.
Thank you, sir.
I would just like to quickly chip in this. I came a bit late, so I didn't meet the beginning of your presentation. And I was wondering if data treatments and cleaning was part of what was introduced.
Okay.
And then the corresponding transformation methods. I don't know if I know this is a very short period that may not have permitted you to speak about that, but
yeah.
I think it's a very good aspect that people need to know that you won't just go straight away to analyzing your data without inspecting, do some treatment, and then cleaning, and then the corresponding transformation for each of those. If talking about linearity, homoscedasticity, and a few other things and things like that. So that may be another thing that's another time that you may the COR may permit you to come over again to speak to us. Thank you, sir.
Okay. Oh,
thank you so much,
thank you very for this. Yes, it's very germane. Yeah, we didn't really talk about it, but I mentioned it when I was discussing the principles of experimentation, that the data must be normally distributed. If it is not normally distributed, you have to pre-process, or you have to clean up the data, or you have to do transformation. And we have different types of transformation system depending on the type of data you are collecting, whether it is binomial data or it is the data that is on counts or scales or scores. and the like. So the time does not permit me, just like you have said, but this can be considered in subsequent presentation. Thank you, sir. Thank you very much.
Thank you so much. Toby, over to you.
Thank you very much, Prof, for this very insightful session and for
Thank you very much.
I'm sure we would want to add 2 extra cubes of sugar to this half, the full cup now, if you permit us. Yeah.
Okay,
and
sir.
I would like to say a big thank you to everyone for joining today's session. To those that asked about the recording is available on the Central Office of Research's YouTube, and the link will be in the chat box in a bit. We'd like to say a big thank you for going over time. It's usually from 8 to 9, and we hope to see you next week, same time and same place. Thank you. And any closing words, Prof, Director, and Divsi, sir?
Yeah, just to let people know that this an ongoing discussion. We may have to ask him to come back and do more explanation on some of those things that he has presented. So let's next week is another time. We'll bring another presenter for your Friday morning coffee. So thank you so much. Maybe my DVC, want to say something to close.
Yeah, thank you. We are 2 months overboard. Again, just to thank everyone for joining and to again appreciate the presenter for this. Thanks for the excellent work. And again, I want to appreciate those who have also made comments to this. So as the director rightly mentioned, by the grace of God, we again meet next week, Friday. Thank you all. God bless.
Thank you very much, sir.
Thank you, everybody.